because transcription is a very, you have seen already, I've shown you already all these kind of guides. And I'm just talking about general transcription factors. Then I'm adding this one, I'm adding this one. Then we'll talk about transcription factors. Then we'll talk about co-regulators. So you see that when you transcribe a gene, you get the involvement of probably hundreds of proteins. Hundreds. The amount of each of them have to be somehow controlled. Because if you put too much of something, sometimes it is a problem for the other one. Or if one is limiting, you have a rate limiting protein, the transcription will not work as well. Here the problem is that when you make, let's say this is the transcriptional activity, it's determined by the amount of RNA that are produced per minute. And here, the more you add of anything, any transcription activator, it can be a general transcription factor, it can be a transcription factor, it can be a co-activator. If you add more, transcription will increase, meaning that this one was limited. Okay, so if you put everything but, let's say, 2F2A, it won't work. Then if you add a little bit, it will work a little bit. And then you add 2 times more, it will work 2 times more. And the more you put, and the more the transcription will work. Until you reach a plateau. Very often you reach a plateau. Why do you reach a plateau? Because now this protein or this complex or whatever you add can be a co-activator is no more limiting. There is plenty of it. So let's say here, when you reach a plateau, it's something else that is limiting the reaction. So maybe it's the transcription factor that is limiting. And so you don't get more transcription. If you would like to relieve this plateau and get a new scope like that, you will have to add extra of these limiting parameters, okay? But what we have seen also is that you continue adding this particular stuff, it goes down. Why it goes down is because the squelching. It means very often one partner will titrate out the reaction important component. So let's say TF2A interact with TF2B, okay, whatever. If you put too much, what happens is that 2F2A will bind to F2B in the solution, not on the promoter. So TF2B will no longer be available for transcription. So the transcription here goes down because you titrate out one parameter. Then you have chromatin remodeling factors. These are all kind, it's a huge group where you have a lot of protein complex that can unwind, trigger, the modification of the structure of the DNA. As I told you, the structure of DNA is a brake for transcription. So you have special enzymes, special complex that helps to unwind, to open the DNA. And this is, that works with all kinds of genes. That's why we put them in the general transcription factors. And again, those ones do not bind DNA. Here you have the important, because as I told you, they have been called general transcription factors. That in the 60s. In the 60s, we were not doing knockout. So people thought that these genes were important for all the genes. Obviously, in that time, we were not doing ChIP-seq as well. So we didn't have a picture of the whole transcription. We had maybe 10 or 15, 50 different genes, and when we look at them, it looks like it was important for all of them. But now when we take a yeast, let's say, and you make a knockout of LPB1, which is a component of the RNA pol 2, 100% of the yeast die. Why? Because RNA pol 2 is the enzyme, the only one that makes transcription of mRNA. So there is no redundancy. You eliminate RNA pol 2, there is no transcription. No transcription can be sure that the yeast will die very shortly. But if you do that with a bunch of them, you see that even sometimes 16%, 3%, only 3% of the yeast will die. Meaning that doesn't seem to be so important. I mean, there is another protein that can replace CRB10. Okay? That doesn't mean, and very importantly, in a knockout, when you see that something else can replace, it doesn't mean that SRB10 is not actually doing the job in the normal condition. Okay? When you do a knockout, you eliminate the protein and you see what happens. Let's say the yeast survived. Well, it survived because maybe something else is taking over. But taking over doesn't mean that it actually does the job when the other one is present. Maybe this one is a lot more abundant, does the job. When you remove this one, someone else is able to replace. Maybe the health of this yeast, even if they survive, they are not as fit as a normal yeast. But you don't see that if you don't challenge the yeast with another condition. Then transcription factors. Transcription factors, as I told you by definition, they bind DNA. So they have always one domain, at least. It's a DNA binding domain, or DBD. All transcription factors have at least one, sometimes even two, DBD. They have, most of the time, all the domain, but this one is critical. They work synergistically. It means that they are making a lot of interaction together. So very often on one promoter or one enhancer, you never find a single transcription factor. You find 2, 3, 4, 5. And it's the combination of all of them that makes transcription so specific to a particular cell, to a particular condition, at a particular time. Then co-regulators, as I told you, definition, they do not bind DNA, so they have to be recruited through protein-protein interaction. Since they do not bind DNA, they have to be recruited by something else, and the something else is the protein. Very often they are recruited by the transcription factors, because the transcription factors, they do not have enzymatic activity by themselves. They are just landmark to recruit the good enzyme at the correct position. And then in the co-regulators, you have two big families. Okay? You have the one that acts on the DNA structure. As I told you, sometimes you have to unwind, you have to open, you have to cut, you have to leave. All this kind of enzymatic activity. All of these activities require ATP hydrolysis. Okay? So the energy is provided by ATP, and then you turn the DNA. Or sometimes you cut, you leave. Then the other bunch, the other group of proteins, they act on the composition of the DNA or on the protein composition. So this is dependent of ATP. So this is, as an example, acetylation of the histone, methylation, phosphorylation, ubiquitination, this kind of modification. These are chemical, these are physical modifications. Both of them are critical, very important. Okay? How the synthesis is initiated. As I told you, at some point, you have to find the beginning of a gene. Okay, determine what is the +1, the position. This is done, obviously, through sequence recognition. Okay? Meaning that on the DNA, you have sequences that allow the protein to bind specifically at that position. The most well-known is the TATA sequence, which is a TA, which is roughly -25, -25, -30. So upstream of a +1. Why it is the most famous is because it was the easiest to find. It is present in a vast majority of genes. I mean, a bunch of them have a TATA sequence. So when we had 20, 30 genes and we compared them, it was very easy to find a TATA box with your eye. Because first it's very simple, it's TATA, and it's located always at the same position. So if you align a bunch of +1, you will see TATA always at the same position. So even with no computer, it was easy to find. Then later on, we found other sequences, because as I told you, not all of them has a tata. So we knew already that there should be something else, because if there is no tata, you require a sequence to allow the TBP and all the pre-initiation complex to form at the good position, okay, on the right spot. And so finally, we find, let's say, E and R. That comes later. But you see the sequence, the pyrimidine. So it's very difficult to find this with your eyes. And the sequence is not perfect. It can be downstream, can be upstream, 10 per 20. So if you don't have a computer, you will never be able to do that, to find it by yourself. Then we find the DPE, which is already downstream, we call it downstream promoter element. So sometimes also we have shown now that some of them have several ones, like INR and a DPE. The more sequences you have, the stronger the recruitment at the +1. Okay? Most of the genes have a very precise +1. So if you take a cell, extract RNA, look all the 5', they will always start with the same base, let's say a C, all of them. But some RNA is not so perfect. Sometimes it can be to -2, +2. You see, you have like a flottement, I don't know how you say that in English, but no, not very conserved. And this is because they say there is only a DPE. So DPE is not very strong sequence, there is just one. So the +1 is not able to bind very strongly, so it moves a little bit. If you have a tata plus an ENR, very strong binding, +1 very precise. You have two models for how initiation really occurs, the pre-initiation and initiation start. The first model came from biochemical analysis in the late 80s. Here we knew already these proteins complex were purified, and the kind of experiment that were done, it's say, I had this one, does it bind? No. Okay? If you put TF2A by itself, it won't bind DNA. You absolutely need to add first the D. But if you put D and then you add the A, it will bind. And then if you add B, it will bind also. This is why it has been called a sequential model. Sequential meaning that every single step requires the step before. If you don't have it, it won't work. Through footprint kind experiment, they have been also to like organize relative to the, this is the, when you see a promoter, you always see an arrow like that. This is the +1, the position, and the arrow indicates the orientation of the gene. Because all the chromosomes always on one direction, okay? But transcription can start on the plus strand or on the minus strand. So when you have a gene, that you look on you see a gene, it will say chromosome 12, blah blah blah, dash, blah blah blah, minus or plus, because it can go this way or this way. Okay, so here we put it this way. So this is +1, then you have a plus, blah blah blah, and then -10, -100, etc. And here you see that the A is upstream of tier 2D, and B is upstream of tier 2T. So this has been shown by biochemical analysis, footprint, this kind of experiment. And that, even today, has been confirmed. Obviously, we have more fancy experiments. Now we can do phosphor linkage. So you have amino acid with a photoreactive amino acid, and then you make a laser on the gene, and so the protein will be linked to the DNA exactly what it is. So now we can even tell you that, let's say, to A goes from -110 to -35. We have very precise location of all this kind of complex and protein. So this is absolutely true. But, and here you have the role of each transcription factors. But then, later on, you see in 1905, it has been shown like a year before in the yeast, and that was shown in the eukaryotic cell in mammals. This was in rat. That actually you have, because in this model, consequence of this is that you don't find binding of these complex except on DNA. Okay? If they are linked together, it's because they are linked on the DNA. But what has been shown is that if you take an antibody against, I don't remember, it was against the first original experiment with CDK7, which is a subunit from TF2H. You make a co-IP, and you immunoprecipitate this protein. What happens is that you can precipitate the whole complex TF2H, so that was kind of expected. Okay, you take one subunit. If your condition of IP are mild, you bring down everything. But they were able to bring down also TF2F, RNA pol 2, a whole bunch of protein and TAF, general transcription factor. That was in opposition of the previous hypothesis where all the complex bind onto the DNA. So the whole paper, it's a cell paper, was to prove that this exists in the nucleoplasm outside the DNA as a complex nolo-enzyme. What is a nolo-enzyme? It means that if you take this, you purify the whole complex, and then you add DNA to that, it will start transcription right away. You don't need the sequential build-up, okay, like in the sequential model. So this is totally opposite. And then, when they publish that, people say it's impossible. So they try to, and yes, it was true. It has been proven in a lot of different labs, on different promoter, different species, mouse, human, rat, etc. What is different is that depending on the lab, you don't have the same component each time. Sometimes someone is missing one subunit, the other lab is missing something else. And that is very easily explainable because every lab uses different buffer, condition of precipitation, and so that you can remove some interaction and not the other one, depending, okay, this guy used potassium, the other guy used sodium, you will preserve all the interaction, but not others. So here you have two different hypotheses, and as today, both are still proven. I mean, we have evidence for each single model, but by themselves they are like incompatible. It's light, the nature of light, it's a corpuscle and a wave. Here has the explanation to put all these together. You have several explanations. One explanation is that you have a sequential model on some genes, but holoenzyme is required for some categories of genes. That's still possible. Another explanation is that the holoenzyme is very important for reinitiation, where the sequential model is only for initiation. So what is reinitiation? Is that when I mentioned that after initiation you have elongation, meaning that the RNA pol 2 leaves the promoter and go to transcribe the whole gene. When it does that, I didn't tell you, but it separates from all the general transcription factors, except with TF2F, which go with it. But all the other guys are left behind, and we thought they will come out. But actually, what we have noticed is that the second round of transcription is a lot faster than the first one. So the assembly is very long, but as soon as you have assemble, it's very easy to transcribe again and transcribe again. So some people believe that the holoenzyme is in equilibrium with the enzyme on the initiation. So when the pol2 leaves, you have a PIC, an incomplete PIC, which is in equilibrium with the holoenzyme. So this is a way to replace the incomplete PIC with a complete PIC.